All right, I'm back for general metabolism part two, and here um, we'll lay out the same metabolic pathways more generally that the carbohydrates use, but now we want to show how lipids and proteins can funnel energy into, well, intermediates into the energy producing pathways that we've already discussed. So just to reiterate, glycogen, again, stored only largely in skeletal muscle and liver. Glucose, which again, of course, can be intracellular, or we think of it also being distributed through the circulation, released from the liver stored glycogen. The skeletal muscle does not release carbohydrates for other cells to use. It hoards it for itself. Glucose to pyruvate. Right, remember that was glycolysis. We can go backwards and forwards to lactate. We have key production of ATP here, anaerobically, right? Because that's glycolytic. Substrate level phosphorylation within glycolysis, and then the pyruvate can enter mitochondria. Drawing this a little bit differently because I don't need the electron transport chain now. I mean, we do, but I don't need to show it. And so we've got our pyruvate being taken up across the membranes of the, remember we're going to give off some CO2. We're going to end up with an acetyl group attached to a coenzyme, entering, it's kind of squashed down here, Krebs cycle. Right? Okay, so that was our, and then again, of course, our hydrogen atoms are coming off of here on NADH and FADH2 to the electron transport chain, which you want to put a couple blobs over here in the membrane as our system of cytochromes, our proton gradient. We lost our outer membrane, there it is. Okay, you get the idea. So now what I want to do is look at lipids and proteins and how they funnel in here. And so let's start with lipids. Well, our primary lipid used for energy is triglycerides. That's how we store lipids in fat tissue. And they can be broken up into di and monoglycerides and free fatty acids. But the basic backbone of that particular molecule is a 3-carbon glycerol molecule linked to fatty acid chains. Right, so this zigzag line here has got a carbon at every bend. And as it's shown there, it's saturated with hydrogens. If we start adding double bonds, then we have to have a double bond. The carbon would have to give up a bond with hydrogen, so it's no longer saturated. So you have unsaturated fatty acids. You got a couple double bonds in different locations. But anyway, so these are our 3 fatty acids attached to our glycerol molecule. And so what the enzymes do in this process is they cleave the fatty acids from the glycerol. And so these guys would actually, instead of erasing it, I'm just going to sort of like, well, erase the whole thing. I mean, I'm just going to create a gap here, right? And now we have glycerol plus 3 fatty acids. And so we've got a 3-carbon molecule, and we've got 2 multiple carbon molecules. Fatty acid chains can be upwards of 26, 28 carbons long. Not a lot more shorter than that, but they can be fairly long. And so a lot of stored energy in the fatty acids. Now, the glycerol is a 3-carbon, so that guy can funnel in as pyruvate. And again, then we could go south with it and make a tiny amount of acetyl-CoA, well, a single acetyl-CoA, right? It's a 3-carbon to a 2-carbon, giving off the CO2. But remember, we could also go north with it, right? So this is also a possibility when we are in need of glucose. So we can take fats to make glucose through gluconeogenesis when needed. We'll come back to that when we bring the hormones into play. The fatty acids are cleaved, and by the way, this happens down here in the matrix of the mitochondria, but I don't have room for it. But these fatty acids are cleaved 2 carbons at a time to produce the acetyl groups, right? So I'm going to have this funnel into here, and that reaction is called, and this is more complex than just chopping those 2 carbon groups off, but that's called beta oxidation. Right, so beta oxidation cleaves, actually also produces NADH, I believe, and maybe even FADH2, blanking on that one right at the moment. Anyway, but we're going to create, as far as the metabolism of the fatty acid, we're going to cleave that to form acetyl groups, which can funnel into the Krebs cycle. So again, lots and lots of acetyl groups per fatty acid to fuel the Krebs cycle. Now we could also go backwards with this, right? In times of excess, or let's say we take in a lot of glucose in our diet, right? American diet is very high in high glycemic carbs. And so excess glucose into here to acetyl groups, guess what? We can take those excess acetyl groups and run them north to form free fatty acids and store them as triglycerides, right? So a high carbohydrate diet actually tends to make you fatter than a high fat diet, or at least similarly so, given sort of equal caloric content. And part of the reason is that the high carbohydrate diet stimulates insulin much more strongly. Glucose stimulates insulin much more strongly than other nutrient types, and insulin is very anabolic to fat production. So anyway, we've got our fats funneling in through pyruvate as well as into the acetyl groups. And key point here, everything is south of glycolysis, right? So we don't get any of these ATP. We get these ATP and we get these ATP. And again, most of them over here. I'm just going to write it here because I don't have my ATP synthase protein shown. And again, the mitochondria does not function in the absence of oxygen, right? We need our final electron acceptor over here, which is oxygen. So if we become anaerobic, then this doesn't do anything, and you're left solely with glycolysis and this substrate level phosphate. This is actually substrate level 2, but you cannot do this in the absence of oxygen either, and I'll point out why in a second. And so anaerobically, you're left with glycolytic ATP production, right? And it means you can only use carbohydrates anaerobically. You cannot use fats, and I'll show you in a moment, you cannot use amino acids from proteins anaerobically. So when you see at the gym, right, an exercise machine that says the fat burning zone, and it gives you a level of intensity, that level of intensity is matched to, they use heart rate as an indicator of level of intensity, but it's matched to aerobic activity, right? It's sort of like maximum aerobic activity without becoming too anaerobic. Because again, once you become anaerobic, are you burning fat for energy? No, right? And so the whole point is it's the fat burning zone because it's both aerobic, but also high enough intensity to burn more calories, but not so high that you become O2 limited and start to do more anaerobic metabolism. Okay. Proteins. I guess I can put the name on it, right? So this is breaking these fats down. Lipolysis, right? Lipolysis, if you will, right, is fat breakdown. And so cleaving the fatty acids, that would be a lipolysis. Beta oxidation is really a kind of a lipolytic function. The arrow going the other way would be lipogenesis, right? Sticking the fatty acids onto the glycerol. That would be lipogenesis. Again, going the opposite way, building a molecule as opposed to breaking it down. If we go back to proteins, then we can go back and forth to their monomer components, which are amino acids. Right? And yes, while there were a variety of different fatty acids, you can still snip them at, even if they have double bonds in them, or if they have different numbers of carbons. You're still snipping them into 2 carbon groups. You're still snipping it into acyl groups, right? Whereas amino acids come in at least 20 different inherent structures, with the R group being the variant component there. What we don't need, what we don't see anywhere in here, is the amino group. There are no nitrogens playing a role in here. And so we're going to take the amino acid and we're going to sort of spur off the amino group. Well, the amino group, when it's on the amino acid, is NH2. When it becomes free, it picks up that extra, nitrogen is a valence of 3, right? And so it's going to pick up another free proton from the solution, from water, and become NH3, which is ammonia. We'll process that in a second. That's going to be a waste product. What we're left with is called a keto acid. I'm trying to get out of the way here, right? And the keto acid is basically the amino acid minus amino group, and so there is a whole variety of 20 different potential keto acid structures. And so they can be funneled in here in a whole variety of locations, and so we're going to call them Ka for short. Right? Now, before we follow those guys up, let's follow the ammonia. And actually, you can actually pick up an extra, form an ion, a cation, pick up an extra proton and form ammonium ion, which is NH4+. So, okay, either one of them toxic. You got to get rid of it to avoid destabilizing proteins and all that kind of stuff. And so, we can excrete a little bit of it. If you produce a lot at once, you start to metabolize a lot of protein for ATP production. As you'll see, I'm going to follow in here. You'll produce a lot of ammonia. It'll actually emanate from the skin. You'll breathe out the ammonia. It smells like ammonia. You have to go to pretty extreme lengths to metabolize protein that quickly and that sort of extensively to produce ammonia so fast that you start to smell like ammonia. But it can happen during exercise. Sort of extreme exercise when you run out of stored carbohydrates and start to mobilize more protein for energy. Not a good place to be, typically. Normally, we're making this anyway, just not so dramatically. And so what we do is we detoxify it to a degree because we can't excrete it fast enough by converting it into urea at the liver. And so we take NH3, we combine it with bicarbonate ion, and we form urea. And then we excrete the urea in urine. It's just a lot less toxic, and so we can sort of carry it around for a longer period of time without it sort of creating havoc. All right, so where are keto acids going to funnel in? Well, some of them have structure where they could funnel in as pyruvate, again, with modification. Funnel in as pyruvate and then be used for gluconeogenesis, so we can use protein or amino acids from protein to make new glucose. Many of them will funnel in here as ketogenic amino acids, right? And so we can form acetyl groups, and then others will funnel in as other different intermediates within the Krebs cycle based upon their structure. So again, we're getting our carbon from protein into the system, but because of the differences in structure, it can funnel in at a variety of different locations. Yes, we can be synthetic with it, right? We can here we're being biosynthetic with the amino acids. Here we're being biosynthetic with the carbons from those amino acids to form pyruvate. And if we're going to go north here instead of south, we can form new glucose with it, which again, a lot of cells like to use glucose for energy. But a lot of those amino acids and the keto acids that are derived from the amino acids are funneling into the Krebs cycle. And then we're going to get the hydrogen atoms out of their electrons with the protons, out of the Krebs cycle to fuel oxidative phosphorylation, development, production of the proton gradient, and all that business that we talked about in the previous video. Now, keep in mind, again, the keto acids are funneling in south of glycolysis, which means they require, again, unless we're being biosynthetic, right? That's the opposite direction. But being catabolic, they are funneling in at pyruvate or beyond, which means they are only used aerobically for ATP production. Now, I don't think I really fully explained that idea of why the Krebs cycle shuts down just because you don't have oxygen over here. Well, the idea is if oxygen is no longer limiting in terms of being an electron acceptor, then the cytochromes can no longer pass more electrons to that final cytochrome because it can't get rid of them fast enough, which means it sort of backs up the electron transport chain. Well, remember, where were the electrons coming from? They were coming from the NADH and the FADH out of the Krebs cycle, which means the NADH and FADH2 are stuck as that, as NADH, FADH2. They cannot be reconverted to NAD plus and FAD. And so the Krebs cycle then halts because we can't get any more hydrogen atoms out of the carbon groups that we're adding in, right, from these other sources. And so this shuts down, right? And you're left with, in terms of ATP production, only with glucose to pyruvate and this very small fractional extraction of energy. Can it be very fast? I said this in the last video. Yes, it can. It's actually more efficient, it has more power, quote unquote, than aerobic metabolism, despite its dramatically lower efficiency. Are you going to blow through a lot of stored carbohydrates very quickly if you choose, quote unquote, metabolically choose to run solely on glycolysis? Yes, you will, right? So yes, it can provide a lot of ATV very quickly. You're also going to run through your energy stores very quickly, right? So it is non-optimal for prolonged energy supply. We sort of do it as a way to backstop being either anaerobic, which again is fairly uncommon, or requiring ATP to be produced much faster than we can deliver it aerobically, right? Again, because this is a higher power ATP per second system than the aerobic pathway. The aerobic pathway just has too many more reactions in it. It's just a slower mechanism than this simple substrate level phosphorylation. Okay, we've got our fats worked into the metabolism, catabolic pathways, along with some anabolic ones, right? Going backwards to our larger molecules. Got the fats in, got the amino acids in. I think all we need to do now is come back with the hormonal control of where they sort of fit into the making and breaking of these molecules, stimulating the catabolic pathways or the anabolic pathways.